Imagine the chutzpah it takes to think one can fabricate foods like milk, fish, and even steak in a laboratory versus in nature. And it is no surprise one of the world leaders of this chutzpah is Israel. And in Israel, one of the leaders is Hebrew University. In fact, food technology has become a hot area of investment worldwide, with the US as the number one source of funding and Israel as number two. Hello, everyone. Thank you for joining us. And welcome to today's webinar, the third in a series we call The Future of Food. Today's topic is growing seafood in the lab, a way to feed humanity and protect our oceans. One of the things that many of us find so exciting about Hebrew University is that researchers at HU are consistently finding innovative solutions to some of the world's biggest problems, not just in the lab, but in actual commercial enterprises. Today, we have the pleasure of featuring one of those companies. My name is David Riccinati, and I am a proud member of the AFHU's Western Region and National Boards. I'm honored to welcome you to this program featuring Professor Eckelby and the company he co-founded, 4C Foods. As many of you know, food technology is just one example of the so many innovations that have come out of Hebrew University since its founding in 1925, 33 years before the creation of the State of Israel. Since then, HU faculty and alumni have been recognized in so many ways. 8 Nobel Prizes, field medals, Turing Awards, 294 Israel Prizes, 10,000 patents, and powerfully 130 plus spin-off companies. And today, Hebrew University is ranked in the top 5% of universities worldwide. Before jumping into our program, I wanted to take a moment to remind you all about Hebrew University's We Are One campaign. The We Are One campaign, established on October 9th, 2023, continues to support the HU community through the provision of mental health services for all who need help, including students and faculty members from Southern Israel and those returning to campus from active military duty. This all gets manifested in 3 initiatives the campaign is focused on: mental health and resilience, Arabs and Jews building a shared society, and strengthening the southern city of Ofakim. In addition, We Are One is funding unique scholarships for the 5,000 plus reservists who have begun rotating out of active duty and returning to the university. We will share the link for further information in the chat, and we are grateful for any donation that you would consider. Now, to further introduce our speaker today, Dr. Eckelby has won numerous grants and awards. Many of these I can barely even pronounce. They're so impressive. But they include the Z-Chi Prize for Discovery and Medical Research, a prestigious ERC Consolidator Grant, and the EMBO Young Investigator Award. And that is just to name a few. He is also an elected board member of the Israeli Society of Developmental Biology, which exchanges information not just within Israel, but also internationally. As a professor at the School of Medicine at Hebrew University, Professor Eckelby established the zebrafish ovary as a new model system to study the mechanisms of ovary development and early egg production. His research resulted in breakthroughs that have contributed to our understanding of the earliest stages of egg production, generating knowledge that is directly relevant to human reproduction. Now, you think that's where the fish part comes from, but no, Dr. Eckelby is a scuba diver who grew up by the sea. He has a real love for the oceans that gave him the idea of how the work that he does in the lab could help humanity solve one of our world's most pressing issues: climate change and food insecurity. Dr. Eckelby combined his developmental biology knowledge with this concern for our oceans to start 4C Foods that has a unique technology to make the world's first cultivated eel meat. The eel is an endangered species with a high demand for consumption, by the way, a high price, and therefore a perfect showcase for 4C's strategy and vision. It is now my pleasure to turn the presentation over to Dr. Eckelby.
Thank you very much, David, for the very kind introduction. Let me just share my screen with you. All right, can you see my screen? Can you hear me well? That's perfect. So thank you very much again for the kind introduction. And thanks for having me. It's a pleasure to be here today and tell you about our work to grow seafood in the lab and in an attempt to feed humanity while protecting our oceans. Before diving into the story, I'd like to tell you a bit about myself and how I've got into this fascinating, innovating research field. And so I am a developmental biologist. I've dedicated the past 23 years to deciphering mechanisms of embryonic development and tissue formation. And we do this in my lab using a little fish called the zebrafish. Those are the little swimming fish in our facility. And we use them for a multitude of reasons, mostly because they're excellent models for human biology and biology in general. For several reasons. First, they share over 70% of their genome with humans, and they model over 80% of human disease, meaning that the same defect in the same gene leads to the same defects and disease, just like in human genetic disease, serving as an excellent model to investigate those diseases, finding cures, and understanding their mechanisms. And especially relevant for our theme today, it has provided most of the knowledge in the literature about muscle formation and how muscle is developing in the embryos and forming muscle tissues. We have developed several methodologies that allow us to record by advanced live microscopy living cells within intact tissues, and not just living cells, but actually in intracellular resolution, looking at various intracellular features here in real time as they occur within the tissue. We develop methodologies to analyze those structures, cellular and intracellular structures, in 3 dimensions, including by deep learning automated techniques. And we develop methodologies to isolate specific cells for their genomic studies. Altogether, providing us with our holistic approach to understand tissue formation. And since joining the Hebrew University in fall 2017, I established an outstanding group of international students and staff. We have secured impressive funding, have made important discoveries in our own research, and gained much recognition internationally. So we're doing pretty well, doing what we do. However, at some point, using our research, I realized that I can exploit my research to expand the breadth of my contribution to society. And I thought that I could do this in the realm of my other passion, in addition to research, which is our oceans and seas. So as David mentioned, I grew up by the Mediterranean Sea. This was my home, 200 meters or 600 feet from the waterline. I fell in love in the reefs of Fiji 22 years ago. This is a traditional kava ceremony that they threw for me. This is one day before my birthday. They're the kindest people in the world live in those islands. And these days I'm trying to pass the passion to the next generation. And the oceans, they're not just beautiful to watch and experience. They actually comprise the major and essential buffer for Earth's climate and ecosystems. Unfortunately, ocean habitats are deteriorating, and they deteriorate mostly because of overfishing. According to the UN, currently a third of global fish stocks have been depleted. With global demand for seafood, it is expected to almost double by 2050. Only under 7% of all fisheries are being fished at under or below sustainable limits, and destructive fishing damages the aquatic ecosystems. The UN issued the SDG 14 for life below water for these reasons, aiming to conserve and sustainably use our ocean, seas, and marine resources for sustainable development. But if current trends remain, ocean ecosystems will collapse soon. And so there is an urgent need for alternative resource of seafood and fish for human feeding. One such alternative is by aquaculture. Excuse me. However, aquaculture also has tremendous implications on our ecosystems and our health. It generates a lot of pollution from fish feed and their derived waste, pesticides, chemicals. They spread non-native disease and parasites. Fish escapes to non-habitable ecosystems, and they have high water footprints. They're also bad for us. It's also bad for us. They involve a lot of industrially produced feed. They contain chemical treatments, and a lot of antibiotics and hormones are being used treating those fish. And so we need another alternative. And the other alternative comes with the promise of cultivated seafood, which really offers a solution for the growing demand for seafood, for humanity feeding on seafood. It has major advantages. It is similar to wild catch. It has consistent quality because the production process is controlled. It is clean, pollution-free, and sustainably forms a sustainable mean of seafood production. So that's very promising. However, you may have heard recently about some concerns regarding the promise of cultivated meat, and this has been raising two major concerns regarding first the ability of mass production of these products, and second, the price of these products. And these two concerns comes from the very complex and inefficient process of making those cultivated meats. So common practices right now use cells that are extracted from fish, adult fish. Those are cells that are never supposed to make muscle naturally. They're being forced to make muscle by different combinations of media, a lot of growth factors that are also very expensive, to force them to proliferate and to make muscle tissue. Those cells are then being put on a scaffold to support their growth and muscle differentiation before being transferred to bioreactors to make them in higher masses and forming a fillet of meat. Now, again, this is a very complicated and inefficient production line, which results in the expensive, unscalable solution. And so on this background, I founded ForeSea with a mission or vision to nourish the world while securing our ocean's health and wealth. I teamed up with our outstanding partners, our CEO, Roy Nir, who has vast experience as an executive in the biotech and food tech industry. Dr. Moriah Shimoni, our CTO, an incredible entrepreneur who has led the work in FutureMeat, and Professor Iftach Nachman from Tel Aviv University, a brilliant developmental biologist, sorry. And together we came up with a unique disruptive technology to produce seafood meat similarly to the way nature does. And this is a very important message here, similarly to the way nature does. Because our technology relies on the fact that the muscle tissue that makes a fillet of meat is naturally formed within the developing embryo. And the developing embryo can be seen as a self-assembly unit for muscle production. It has all the instructions to make muscle, all the information is intrinsically provided by the embryo without the need for external cues. And so we sought to exploit this natural process for our technology. And what we do differently is that we extract cells from fish embryos, the real cells, natural cells that are supposed to naturally make muscle and differentiate into muscle. And then we use our proprietary organoid technology. Those are small masses of cells that mimic the embryos. They mimic the embryos, including its ability to form muscle in a very natural manner. And then we have to, again, use bioreactors to expand these and make our fillet of meat. Now, this technology allows us to use much reduced growth factors. It eliminates the need for scaffold. It's much closer to nature. It requires no specialized immortalization. These are the natural embryonic stem cells that are being used, and it has much better scalability. And so the first stage in our technology is to isolate those cells from embryos. And here you see, and here we use the zebrafish in the beginning. These are little zebrafish embryos that you see here. Those special cells, the embryonic stem cells, are right here where those arrows point to. We have the knowledge to identify them and isolate them, culture them. They form the morphology of true stem cells. We can then subculture them and transfer them again. They form the same morphology. And we're also able to show that they exhibit the molecular signature that is really characteristic for true stem cells. Now, why zebrafish, right? So most edible fish species cannot really be bred in captivity in a normal lab space. Their genome is not appropriately annotated, and there's no really experimental tools available, and neither is knowledge for their study. In contrast, zebrafish has been used in research for over 40 years now. It is easy to breed in captivity, as you saw in the video earlier. It's small and transparent. We can see through the organs and cells. It has excellent genome annotations, a wealth of experimental tools, and vast knowledge. And as I mentioned earlier, most of the knowledge on muscle formation comes from zebrafish studies today. And so by using the zebrafish, we're harnessing over 4 years of technology to a completely new need. And here you can see some other advantages. This is a living, developing zebrafish embryo. These are the cells that are being divided during early embryonic development, from 1 to 2 to 4, and so on, cells, until at this stage, right here, close to 1,000 embryonic stem cells per embryo are available. We can get yields of dozens of embryos per cross that we're setting up, and thousands of embryos per day. So very efficient. And the process is very similar across species. The process of muscle development, the same cellular processes, the same molecular regulation takes place between zebrafish and other fishes, and also between zebrafish and other animals. What you see here are a mouse embryo and a zebrafish embryo. Here is our mouse embryo head and tail and the back. Here is our zebrafish embryo head, tail, and back. And you notice the small packages here where the arrows point. I hope you can see my pointer. Those little packages are called somites. Doesn't really matter, but these are the units that form muscle, bone, and fat in the embryo. And you can see these are very similar. And in fact, if you look at a fish, reptile, bird, or human embryos, they all form muscle in the same way within those packages. And so whatever we learn from zebrafish, we can extrapolate in a very seamless manner to other fish as well, making not just a technology for zebrafish, but actually a complete technology to cover all fish. And so back to our approach, we show that we are able to extract the cells, but now I'd like to show you those organoids that we're able to make. And here you can see those little organoids. What you see here is this mass of tissues mimicking those little packages in the embryo. In green It's a molecular marker for a progenitor of muscle cells. Those green cells will make muscle later on. And indeed, later on, by these markers, we can detect proper muscle cells and muscle fibers within those organoids. And this is a patented technology. We have a full proof of concept for organoid technology using our proprietary of cell lines. They have proven capabilities to grow organoids in suspension, which is highly relevant for our purposes. Our protocols show both the ability to differentiate in muscle and fat. It's suitable for the scale of bioreactors, and we can achieve high cell density with those. And so we're basically harnessing the natural embryonic way of tissue formation to make a seamless, straightforward technology that can be transferred to any fish. Again, mimicking the natural way, just like in nature, the embryonic way of making muscle with minimum intervention or artificial enforcements. And our first product is the eel, and the eel is really an exemplary species for our goal of restoring ocean habitats. The eel is an endangered species. The unagi, as it's called, population has declined 90 to 95% in recent decades. It cannot be. It has really high demand. But despite this high demand, because of this decline, the consumption dropped, and it's really considered as a high quality or really high demand, high price fish. And it cannot be bred in captivity because its life cycle is really complex. Those eels in different steps of their life migrate between shallow waters and deep waters across miles of different ocean habitats. And not a single lab to date were able to really breed and close the entire cycle of those fish. And indeed, recently we were able to announce the first cell culture unagi in the world. We teamed up with Chef Katsumi Kazumoto from Saido, a top vegan restaurant in Tokyo, who helped us cook those products. And this has created a lot of enthusiasm by many in the world, which is really exciting. However, we have more than unagi, and we can our cell culture products involve other fish and other dishes, as can be shown here. Now, it's worth noting that this is a very attractive market. It's based on those calculations of the current global worth of this market, and taking into account the current consumption, the expected growth in consumption, and the prediction of alternative seafood taking a share or making a share of 7.5% of all seafood, you come up to a $40 billion market. And so far, I'm really proud to say that we've been able to achieve all our milestones, including our own representative in Japan, and a strategic partnership with the world's largest seafood company, and also Japanese company, Marua Nichiro. And this is also exemplified by or reflects our business model as a B2B2C, where we will distribute the products to local distributors, which will in turn distribute those products into local grocery stores, restaurants, and so on. And excitingly, we're expecting to launch the first product at about Q4 25 to Q1 26. And the reason I'm telling you about this is because I'm really, I take pride by the fact that we're able to couple commercial interest with the high cause of saving endangered species and habitats, which is, I think, the most efficient way to really make a difference. And I hope I was able to convince you today that output from our academic and commercial endeavors will make a real impact. By our technology, hopefully will be able to save many species by reducing fishing de facto. By this, we'll be able to restore oceans habitat, improve our oceans health, and conserve Earth climate and ecosystems. And with that, I'd like to thank you for your attention. I'll be happy to take questions.
Fantastic. I'm impressed. I'm sure everyone else is who's on the line. Taking cell biology, translating it to fish is really quite remarkable. So yes, we have the Q&A open. So let me start with the first question. I thought it was interesting that the first product you showed was in a vegan restaurant in Japan, yet this product is coming from an actual cell. So is this vegan? Is it fish? Is the eel kosher?
Very interesting. So those are really interesting discussions. I think the whole point of being vegan is to avoid consuming live stocks and live fish and live animals. This is by no means a live animal. We are dealing with cells that are kept in the lab. No real animal is being hurt by any way during the process. So this is being welcomed really excitingly and enthusiastically by vegan and vegetarian communities that will be now able to consume fish meat products. I think it's a great way to live by your ideals without too much sacrifice, right? Now you don't need to make a sacrifice. You can enjoy the flavors of meat without really hurting any animal, which I think, and it doesn't require people to sacrifice a lot, which again is the most efficient way to advance any field, right? Without taking too much effort from people. Now, regarding kosher, there are also ongoing discussions. It's not really my domain. However, I think it's being accepted well by religious authorities and rabbis that are now discussing exactly those issues. It certainly raises a discussion and a constructive one.
Sure. But I assume this is not just for vegans. If you are an eel eater today, would you be able to tell a difference between this product that you've created and an eel that came out of the ocean?
So no, this would be exactly the same. And based on some testing that the company has done, and the company has a food engineer and that sees into exactly those processes, you should not be able to tell the difference.
Amazing. So we've got some business questions coming in as no surprise. I can tell we've got some venture capital types potentially on the line. People are asking you questions like unit economics. How far do you have to scale where you can make the eel profitably and sell it at what I assume is just normal market prices? Are you close? Are you a couple of years away? How do you think about that?
Right. So the first launch of the product is supposed to be already in much lower prices. And again, our CTO, Moriah Shimoni, she's an expert exactly on those issues. This is what she has done for FutureMeat, and now is leading also with us. The fact, I mean, already our fundamental technology, reducing the amount of growth factors that are necessary, right? So as I mentioned, by common practices, those cells that are not supposed to make muscle are being forced into making muscle, but by using a lot of growth factors. Those growth factors are really expensive, and you have to use them by gallons and gallons. This is, a big cut of the price is coming from this. And so we expect that we'll be able to cut up to 90% of those already at this stage, just by the fundamental technology.
So does that imply that making fish in this way, or eel to start, would be 90% cheaper than the way everyone else is doing it, or 50% cheaper?
So these are
You can tell we've got some interested parties out there, Professor.
right. That's amazing. The commercial domain is not exactly my domain. Our CEO can answer those questions really well, and I'm happy to connect him with anyone interested in more
I think
precise questions.
I think that's a great idea. If we have anyone who's listening who is interested in the business on the commercial side, if you're open to it, Professor Appleby, let us know through the chat, or sorry, through the Q&A, and we will make sure they get introduced to the company.
Absolutely.
So when do you think EEL will be coming to the United States or Britain or Canada? I think we have representatives from all countries on the line.
Right. So we're first targeting the Japanese naturally because this is considered some of their cultural aspects. But once we're there, I mean, Maruo and Ichiro are the world largest distributor. So I guess really soon after. I don't think we should expect gaps in there.
Got it. So we here in LA can look forward to seeing it in our sushi restaurants, hopefully.
Absolutely. That's the point. That's exactly the point.
Right. So let's talk a little bit about nutrition.
Yes.
So does we're just using the eel that you described, it tastes the same as you had mentioned earlier. What about the nutritional value of the product?
Right. So basically, because it's still made of normal muscle, right? So it maintains all the high quality of muscle with protein level. So just basically naturally it can be the same. But actually I'm saying that it can be the same because it can actually be improved because during the process of making the fillet, you control how much fat to introduce and how much muscle, and you can actually make it even healthier nutrition wise than the natural product.
Of
And
course.
of course, there's a whole diversity and variability in nature. One eel is not necessarily identical to another, while producing this in a lab is very accurately done the same way, promises constant quality in each of the products.
So you could actually make the product, the eel, more nutritious, however we define nutritious, if you wanted to.
We won't artificially change the composition of the muscle cells, right? This is not the idea. But if you wish to make it a little bit more lean or a little bit more fat, this can actually be absolutely can be done.
Amazing. So people are interested, what kind of hurdles, regulatory hurdles, who approves that consumers can actually eat this product and it's safe relative to natural seafood?
Right. So that's another exciting domain, which I'm not an expert on, but I can say that there's already advancement in regulation in both USA and Singapore, and we're actually working. Our representative in the name of FORESEE is working with Japanese and Singaporean authorities to advance the regulation of this. So we're working on all fronts.
Do you get any sleep?
Not too much,
It's a lot of fronts you're working on, Professor. I love it.
That makes it interesting.
So someone's kind of interested. Apparently, there's a big industry of farming fish for animal feed, and so more of a B2B product, an input into something else. Is that relevant here, or is this really more focused on what we consumers would eat?
So we're focused on human consumers, yes.
Okay. Got it. So if you're dreaming now for a second, now FORESEE Foods, eel is out there in the market. It's in the sushi bars of Los Angeles. What fish is next?
Well, we have several of them. That's unfortunately all I can say right now. The company didn't disclose all the other ones, but they're coming up. They're definitely coming up.
Well, you showed some beautiful photos. of a couple what looked quite tasty, if I remember correctly. So I guess we can expect Moroccan fish.
Oh, in terms of, yes, absolutely. The dishes that I showed you were already able to do. The type of fish that's in there, we didn't disclose yet, but there are other fish.
So are you, is it just in your lab, or are there places in Israel where people can actually eat those products
All right,
that
so
you show?
it started by our collaboration with my partners. We started in my lab. Soon we expanded. I mean, we already have a team of 16 people working on our own dedicated site in Rehovot. Very impressive site, highly equipped, top of the notch equipment, and they're working there much faster and harder than anyone can work in an academic lab. So we developed the initial technologies, myself and my partner from the University of Tel Aviv. But those we tech transfer, we transfer our technologies to the company, and they take those and they make them better, and they make them in a much more robust and manner that can with all the commercial potential and impact that the company is expected to. So we're working really closely with a very nice and efficient collaboration between our own academic labs and the company site, who's really taking it to the next level.
Great. And that maybe we'll wrap up here in a couple minutes, maybe just a few more questions.
Sure.
How has Hebrew University helped you in this endeavor?
Oh, so the commercialization company of the university, Zoom, was really key here. So when I first started, playing with the idea, right, when I heard about cultivated meat and made a connection between cultivated meat and our oceans, and coming together as an interface with cultivated fish meat, I reached out to the university and I told them I want to be engaged in such endeavors. And they said great, and they immediately started working on looking for opportunities. And we were very lucky that on parallel, the kitchen, our hub, the hub company that helped us found 4C, was looking for the same thing, and contacted my partner Iftach from Tel Aviv University about his organoid technology, and then they contacted Isum, and Isum said yes, we have the right, just the right person, and they immediately made all the connections and helped us come together and helped us with some of the bureaucracy and the procedures of getting started. Um, um, so yes, this was a match from heaven, I guess. This is just right.
It was a
You know,
shidduch.
yes, absolutely. And we feel lucky, but we feel lucky, but we also have to remember, chance favors the prepared mind, right? So this was like
100%.
just the a good case to show it, I guess.
Luck is preparation times timing, for sure.
Exactly.
Maybe for those people who don't know, could you just quickly describe Yasum to the group as we wrap up
Oh,
here?
sure. So Isum is a company by the Hebrew University of Jerusalem. Its mission is to commercialize technologies that come from academic labs. So laboratories that develop any knowledge or technology that has the potential to be translated into commercialized technology. Isum helps them do exactly that. And establish companies, startups, getting in touch with hubs, and so on.
Great. Okay, I think we're probably down to the end of our questions. There's some very specific questions about what you do, how you do it, costs, growth mediums, and I guess I'm going to say to everybody, if you're really interested in the company and you have some really specific questions, I am sure we can compile a list of you and get you in contact with either Professor Eckelby or the CEO, whatever is appropriate, so you can learn your more detailed questions. And potentially, I'm going to say, even if Professor Eckelby, you're looking for investors in the future, we may even have some on the line. So I hope we can make that kind of shidduch too. So maybe we should wrap up. And again, this Yasum point I think is very important that the professor talked about, is that the Hebrew University has the infrastructure in place to marry innovation coming out of labs with actual scaling for commercial enterprises. And it's a very important part of the Hebrew University ecosystem. So I was very glad to hear that you are a great case study of that in action.
Absolutely.
So I know,
Grateful for them to them for that.
and I'm,
I
go
just
ahead.
would point it out. Feel free if you want to drop my email on the chat box. That's absolutely fine. We are open to communicate with everyone.
Fantastic. And I will maybe leave that to the team, whether it's in the Q&A or the chat, or maybe a follow-up to the webinar on how we get your information out. But I do want to say on behalf of, we had 71 people listening in today. My guess is from many different countries. And I think it just speaks to the importance of this topic and the power of what you have accomplished in a pretty short period of time, taking learnings from one area and applying them to another, I think is what's most exciting to us. So thank you, Professor, for your time this afternoon, this evening. We very much appreciate it. And please all, will be looking forward to Future of Food edition number 4 that will be coming out sometime here in the second or third quarter. Thank you. And I think that wraps us up for today.